Yeah,
Like, I'm trying to understand. Okay, this is like maybe another thing. Right? So when I first start talking about candies, like I can candy. That means we'll jump on her after class. Uh, that's how it goes when somebody has candy. I have a math. So quick catch up. Let's see. Let's start candy class. Culture is winning the costume competition. You're yeah. Oh my god, I love that. I've never seen my dog. Yeah, me and my husband played it together, and did I go set up my ambitions time working, blah blah blah. I love very short, I got a division, 3 and love like that. Like, oh, yeah, for I was really love. But I think that is a goal. And yeah, no, I went to a team and I was like, yeah, I just selected it and out as a goal. And then my husband, we had a house supply unit. Yeah, but yeah, it's kind of. Then we had a son of Tivatino father, who didn't with the stick by himself. Then he hired the maid in Greece. Then he pregnant with the maid, and then he quit. Yeah, so I was like, okay, who am I marrying? Why did I do that to myself? Yeah. Canada, I said like, and that's when I decided if I die before him, I'm going to come back and run. So he never gets married. I said, I don't hold it because I'm too unprofessional. But just so you know, I also like Halloween process. I'm a TA who is a pilot.
yeah. I don't even have the best part of the costume on. Wait, the best part of the costume is the gloves.
Why?
Because they're like, wait, see?
Oh,
Wait, this
oh no, it's amazing. You see, I'm a big fan of Halloween. So when I was like your age and my sister's age, I was dressing up constantly.
okay, I'll just sit down.
You look lovely, and we have a Sims character over there. Uh-huh. Yeah. So no, I really like Halloween, and in fact, in Wyoming, I would buy 3 costumes, like because you can't go to 2 different parties in one costume, right? No, no, you buy it. Depends how many parties you have. If there are two, you buy two. If you have three, you buy three. But you can. No, 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 no. Girl never goes to the party in the same costume. You have to. I have to mentor you a little bit more. That's like a wearing the same dress, same to 2 different parties. No. Yeah. What is wrong with you people? Come on, different dress. That's on Thursdays. Okay, so let's go to the now when we're going back a little bit. Let's talk about how we started this course. We feel which is nitrogen metabolism and control, which is glutamate decader. That's what I want to see. I love it. And please expect the class. There will be again 2 slides. Memorize them like the life event. Because that will be questioned. I adore asking those questions just because it's so easy and you don't have to, you just understand. And that's it. So, is there anyone who has exam later on because who are you how well we started? We're ready, okay. We are going to the battlefield later by 3 to 2 has the quiz. And they don't freak out, they think it will be hard. It won't. It will be very easy. But I freak them out on purpose. So they start. So anyway, good dehydrogen is the first and the most important enzyme that I would like to mention in this class. Glutamate synthase is another one. Both of them alongside of the GOGAT are controlling production of the most important nitrogen forms in our organism, which are amino acids. So glutamate dehydrogenase is, as said here, spread all in all life forms. So we have it, microorganisms have it, and what this enzyme does is simply amination of alpha ketoglutarate in this mixed cycle. So again, you get alpha ketoglutarate, you get amino groups from ammonia, and then you have glutamate dehydrogenase or GDH, and that yields glutamate. So if you want glutamate, you have to have alpha ketoglutarate, and you have to have GDH. What else you have to have? You have to have NADPH, because NADPH, which is reduction equivalent, gets oxidized to NADP+. So what that tells you, this is a reaction that actually spends energy. So it's anabolic reaction. On the other hand, there is an enzyme called glutamine synthase or synthetase, and that enzyme is very characteristic. So everything in this portion of the lecture will be mostly actually dependent on the activation and regulation of GS enzyme. So GS enzyme is literally taking already built glutamate, and again, glutamate is built by GDH, and adds second amino group to yield glutamine. Because if you remember, glutamate and glutamine are two most important nitrogen donors in the cell. Those are amino acids, and glutamine has two amino groups, while glutamate has only one. Glutamine synthase or synthetase is tightly paired with a third enzyme called GOGAT. There is a long name. All what you need to know is GOGAT. Why? So it's paired with GOGAT to yield another glutamate. So again, so a second enzyme that makes glutamate from glutamine. Why we need glutamate? Because that one is the first and the most important nitrogen donor in cell. Glutamine is the second one. So which enzymes are yielding glutamate? GDH and GOGAT. Because they have different routes and they are activated in different stages of the organs. So GS and GOGAT are paired together to yield glutamate as the most important nitrogen. So what is their principle of action? So once when ammonium, which is a gas, enters the cell, it becomes at neutral pH, it becomes ammonium ion. So the first step, this is the first step to build universal nitrogen donor in the cell, which includes getting ammonia in the cell, and ammonia becomes ammonium. To build glutamate, you need to have alpha ketoglutarate from TCA cycle, which will be converted by GDH or glutamate dehydrogenase with a standing of 1 NADPH into the glutamate. Again, this is reaction that consumes one reaction equivalent from alpha ketoglutarate to get this glutamate. So this is all great in normal status of cell. Alpha ketoglutarate, GDH active, and you get glutamate. But what will happen when concentration of ammonia is very low? How will you deal? So that's like a nitrogen starvation. So there is not enough ammonia in cell. So if ammonia in cell is very low, the organism needs to implement activation of second enzyme pair. Actually, the first one is GDH, and this is enzyme pair GS-GOGAT. Why? Because we need to build glutamine. Because glutamine has two amino groups. So now, in that situation, glutamine synthase will literally grab already built glutamate, and again, glutamate was built by GDH, and will add one amino group to build this glutamate. As you can see, this reaction consumes one ATP, so it's energetically intensive reaction, pretty much the same like GDH, just GDH consumes an ADPH. Now you have glutamine, and again, we are now in nitrogen starvation. So from that glutamine, you need to build the most universal amino donor in organism, which is glutamate. So again, glutamine synthetase build glutamine. But now from glutamine, you need to build glutamate, which you will do with a GOGAT enzyme. That's why we say that GOGAT is paired with GS, glutamate synthase. And this is also an energy required and energy intensive reaction because it consumes one NADPH. It oxidizes NADPH to NADPH. The result of that is actually most important nitrogen donor in cell, which is glutamate. Again, this is happening when nitrogen level is low. So, brief summary, again, at the end, this is just a brief summary of first very basic process. GDH, glutamate dehydrogenase. GDH is enough to know. But again, you don't really memorize. Consumes alpha ketoglutarate, spends one NADPH and yields glutamate. Glutamine synthase or GS consumes that glutamate, aminates it at second amino group and spends ATP molecule. And last but not least, GOGAT that consumes now glutamine, deaminates it and spends one NADPH and makes NADP plus to yield glutamate. So these three enzymes are actually working in harmony to provide both important nitrogen donors in cell. So what are then, how those two pathways for nitrogen regulation operate in cell? So why microorganisms don't have only GDH? Well, GDH or glutamate dehydrogenase has very low affinity for ammonia. What that means is that in order to have that first pathway of glutamate dehydrogenase active, you have to have high amount of ammonia because it has very low affinity. If you don't have enough ammonia, GDH won't be active. So when there is a lower level of energy, when cell is starving, there is not enough ammonia, activity of GDH will be low. That means that you have to provide second pathway that will yield more nitrogen so you can get GDH active. And the way how you will get more glutamate and more like active enzyme would be with yielding of glutamate. So when there is a high ammonia, like there is no nitrogen formation, you will get GDH. But when there is a lower level of ammonia or nitrogen starvation, the system that needs to be activated is GS GOGAT. Why? Because GDH has very low affinity for ammonia, and in order to get it activated, you have to have ammonia overload, which of course won't happen when there is low ammonia. So both glutamine and glutamate are derived through GDH and GS GOGAT systems, and those two are two universal nitrogen donors in cell with the fact that glutamate is the first and glutamine is the second. Again, that's important. That's what I want to say. All other amino acids, and we as bacteria also prokaryotes have 20 proteinogenic amino acids. All of them are generated from glutamate and glutamine in different reactions like transformation, adaptation, deamination. So these are three most important reactions that each of those molecules can pass through to yield nitrogen compounds in order. So transformation means like transferring between nitrogen groups. Deamination means removal of nitrogen groups. Racemidation means changing a structure from L to D4. So in summary, there are two enzymatic systems for assimilation of hydrogen, GDH and GS GOGAT, and each of them will be activated depending on nitrogen level. So if you have ammonia present in high amount, you will get activated GDH. If that's because it has low affinity, you lose nitrogen, it will be GS GOGAT because it has higher affinity. So the connection between GS and GOGAT, which is a second pathway, is done for balancing purposes because cell always has to provide the most important nitrogen donor in cell, which is the first one, glutamate. So that's why GS and GOGAT complement together. And this is just a list of amino acids that are built from certain metabolic compounds. And I will tell you exactly which one I insist of knowing. So obviously, you have to know that glutamate is derived from alpha. Arginine, not so much. Like you will. I don't think you will have a hard time there. But I think because arginine, glutamine, they are basic amino acids. So glutamine is derived from glutamate. So arginine is the same. But they are from alpha ketoglutarate. The most important is that oxaloacetate is a precursor for aspartic acid production. Pyruvate from glycolysis is precursor for valine and leucine, and I should add here alanine. 3-phosphoglycerate or 3-PG is a precursor for cysteine. The most important that I really like to ask is the phosphonopyruvate and erythrosyl 4-phosphate. Those two metabolites are the only precursor for these three aromatic amino acids, which is tyrosine, phenylalanine, and wheat. So again, aromatic amino acids are asparaginine from oxaloacetate and glutamine, glutamate, and arginine from MICRB and the hydrophobic amino acids like valine and leucine from pyruvate. So how is regulation done? And this is where the party is. So when cell has a normal level of nitrogen, or it's not up, or it's not target for nitrogen, GDH has basic expression and it's normal expression. So it creates glutamate normally from oxygen. When cell starts starving, obviously GS-GOGAT system needs to be activated to stop out the nitrogen. And there must be tight enzymatic control at various levels, transcriptomic and translational or enzymatic levels. So there are two stages of control of GS-GOGAT that regulate function of these two enzymes in order to balance level of nitrogen. So again, GDH has low affinity for nitrogen, so these genes are constitutively expressed. But when there is low ammonia, GDH concentration and activity will drop significantly. What will happen in low ammonia? That nitrogen stagnation. That means there will be stringent response. You can already guess that when stringent response is activated, you will remember relay makes PP, VPP, PP bind to DKSA, and then that complex PP, DKSA goes down and promotes association with specific sigma factors with RNA polymerase. Sigma factors that will play a role in this one will be sigma 52, but why? Because it's nitrogen. So sigma 70 goes south, sigma 54 comes in, and regulates genes involved in control of nitrogen processes in cells. So that means the GS-GOGAT has to have tight regulation to sense the level of ammonia in the cell. The way how it will sense is through alpha ketoglutarate and glutamine. Because when it's low ammonia, you will have a lot of alpha ketoglutarate. When it's high ammonia, there will be plenty of glutamine. And that's what will regulate GS-GOGAT. GS-GOGAT is very complex enzyme, and it has like 12 subunits in two different planes, 6 in one, 6 in another. And it has on each unit it has place to add AMP or adenylate. So one of the first controls of GS is adenylation at tyrosine rest. So that adenylation, and adenylation comes from ATP. So ATP gets converted to AMP, and then AMP is added to GS. So the place is on each subunit, and there are 12. How that functions? So again, 12 subunits. This is how complex it is. So it's huge. And there are 12 subunits. In each subunit, you have a place for adding AMP to be added. So who is doing adenylation? So here, let's start like this. So when GS needs to be, so why GS-GOGAT needs to be inactivated? It needs to be inactivated because it spends a lot of energy. So if cell is not starving, if there is no need to make glutamine, then GS needs to be inhibited. It needs to be deactivated. So it needs to be controlled. So when cell has a lot of glutamine. There is this enzyme called GLNE, like E enzyme. You will see later we have B and D. So GLNE enzyme has dual function. It has ATase activity and ARase activity, which means it can add and remove adenyl from GS based on the glutamine and alpha ketoglutarate status in cell. So when there is a lot of glutamine in cell, that means cells are not starving for nitrogen, which means the GS should not be active. GDH is fine, there is a lot of ammonia. Then GLNE enzyme will activate its ATA or adenylation activity, complete hydrolysis of ATP, generate AMP, and then add that AMP or adenylate to each of these 12 substances. When GS is adenylated, again, adenylation done by GLNE, and GLNE has ATase and ARase activity, but when there is enough glutamine, ATase activity is promoted. Adenylated enzyme, zero activity. GDH does the job, enough nitrogen, everyone happy. Okay. Now remember GLNE and its ATase activity. Because what will happen when ammonia is very low? That means there will be more alpha ketoglutarate. So that's a signal. And the signal that there is a lot of alpha ketoglutarate and a low glutamine, that is going to be sensed by GLNB. GLNB now binds to GLNE, the one that does adenylation, deadenylation. And you remember how GLNE first had adenyl AT activity, like adds. Now, when GLNB recognizes there is a lot of alpha ketoglutarate and low glutamine, it binds to E and reverts its activity. So now GLNE does not have AT, it has AR or deadenylation activity. Because we say that this is AT is like a bifunctional enzyme. So AMP is removed. When AMP is removed and it's done by GLNE, because GLNE can add and remove, it's bifunctional enzyme that activates glutamine synthase. I told you it's complicated. So to be more complicated, we are introducing another enzyme called GLND. So you have E that has ATase, ARase activity, and you have D and you have B. So what is D? So D is also bifunctional. We have UT and UR. So when there is a lot of glutamine, when now let's say cell built a lot of glutamine when GS was active, that means that level of alpha ketoglutarate is dropping. That glutamine will literally occupy GLND. With GLND occupied with glutamine, GLND activates UR activity, which means it removes uridyl groups from GLND. So again, GLNB is this one that pretty much regulates AT-ARase activity. We talked about that in the previous slide. But that GLNB can be uridylated or deuridylated, like with uracil or without uracil. And the guy who takes care of that is GLND. So when there is a lot of, and then GLNB is naked, reacts with GLNE, GLNE now activates ATase activity and adds AMP to inactivate GS. So how will you know it? So again, we will go in details, but brief. Active GS is adenulated. To be adenulated, you need to have deuridylated GLNB. So it's kind of complicated. Uridylated GLB promotes the rules. So it's like the opposite. That's how I memorize. Again, and what happens when glutamine is low, alpha ketoglutamate is high? There is no glutamine to bind. Yes. Yes. GLNE is the one who does the job. But all those GLNE are just when I'm talking about medical. So I'm just briefly mentioning this because this is short active for the nitrogen cycle, and it's not the end. There is transcription control. So when alpha ketoglutarate is now low, when alpha ketoglutarate is now high and the glutamine is low, GLNT is now free of glutamine, and now it activates UT activity. So adds uridyl groups to GLNB. When GLNB is uridylated, it reacts with GLNE, and GLNE activates adenylyl removal activity, or so it has both AT and AR activity, and removes AMP to activate GS to create more glutamine. So, brief overview. So again, GLNA, I never ask students to know the name of that gene because that's just a gene for glutamine synthase, GLNA. Important is AKAs are ARA. So this is same enzyme, GLNE, that has low activity. The one who has, so don't get confused. So GLND has like URUT activity. B is the one that gets uridylated and deuridylated. So two enzymes with the dual functions are GLNE and GLND. B is the one who gets uridylated. Same how GS gets AMP. So when I ask you a question, don't get confused by that. You won't. Okay. Any questions? Any tears so far? Everyone gets it. I see, oh God, my three grad students have to get that, or I'm going to quit. I see crying, no crying. It's easy. It's easy. It's really easy. We will go in details. Really. So you are there, it's a very enough. Yeah. So it's easy. It's really easy. I mean, and this is not so hard. Like transcription control is actually beautiful. When you integrate everything together, it's cool. So we are going back. So this is what I was talking about GLN, DNB, and E is all about enzymatic control, like adenylation, uridylation. Who gets adenylated? Yeah. Who gets uridylation? GLNB. Who does it? Who does uridylation? GLNB. Easy. There is not like really. Once you get it, it's fantastic. This is the best part of microbial physiology. So let's go back to transcription. So sigma, when sigma 70 gets associated with RNA polymerase, that is happening in normal conditions when there is no starvation. When there is starvation, sigma 70 needs to dissociate from RNA polymerase, and you need to get sigma 54 associated. That is called stringent response. And this is just, I'm not going to repeat this because I said this literally a few minutes ago. How stringent response happening, polymerase activate genes for biosynthesis of amino acids. So this is just reminder of stringent response. And that was your listening question. So when it's nitrogen starvation, sigma 54 has to know that. So it starts transcription of genes for amino acids because apparently we are starving for nitrogen, now we need more amino acids. So this transcriptional control of GL or of glutamine synthase or GF-GOGAT is done through something that is called cytoplasmic two-component system. So we talked a lot two-component system, and usually we talked about the membrane protein. So you have a membrane sensory kinase get alpha phosphorylated which causes phosphorylation of response regulator on aspartate unit, and then that response regulator controls transcription of certain genes. Here you have very similar situation, just it's not in membrane, it is in cytoplasm. And the molecules that are in charge for that are called NTR, B, and C. So NTRB is sensory kinase here, and it gets phosphorylated, like does autophosphorylation on histidine residue based on the level of nitrogen in cell. And when NTRB is phosphorylated, it goes down to another molecule, NTRC, and phosphorylates that one. So the response can it can do the response. It can do transcription of certain genes or amino acids. So basically phosphorylation status of NTR, B, and C is what will decide which gene will be transcribed. And let's go now in details. So NTRB is sensory kinase, autophosphorylation. So autophosphorylation first and NTRB is happening when there is nitrogen saturation, which means high alpha ketoglutarate and low glutamate. Because there is a lot of alpha ketoglutarate can bind to GLNB. What is GLNB? We talked about it. That means GLNB will get uridylated and bind to NTRB. NTRB now gets autophosphorylated in histidine, and that phosphorylated B is going to transfer phosphate to NTRC, and NTRC is now active and starts transcription of genes for more and more glutamine synthase because GS is needed because cell is starved. So phosphorylated NTRB is bound to uridylated GLNB and deadenylated GS. What is happening in seconds? When there is high, this should be high, not how. High glutamine and low alpha ketoglutarate. So again, sensory kinase that sends nitrogen level. Glutamine is high, alpha ketoglutarate is low. So we know that now we know the active. So now NTRB is naked. It's not phosphorylated. So in the same time, GLNB is not uridylated, like deurydylation, and GLNB is now naked. And so naked, it binds to NTRB kinase, and that shuts down something. So NTRB binding to the naked GLNB stimulates additionally N phosphatases. So all additional phosphates are removed from response regulators, NTRC. And when there is no phosphorylation, that means no activation of GS genes, which means GS is what? Adenylated, is the adenylated. So how is that going? GS expression is always little active, so it's not completely shut off. Promoter 1, so this gene for GS, this is gene GLNA. Again, I never asked to memorize that because I never memorized it. So there are two promoters to control that gene. One promoter is regulated with RNA polymerase that binds sigma 70. The other one is regulated with RNA polymerase that has sigma 54. So there are two promoters. So constitutive expression is allowed when alpha ketoglutarate is high and glutamine is lower. NTRC phosphorylated binds and enable control and the increased expression of GLNA or GSG. So this is how promoter looks. In mini summary, before I give you like a all large overview. So transcriptional control, NTRB, NTRC. NTRB senses a high alpha ketoglutarate and low glutamine, that means nitrogen starvation. When it's nitrogen starvation, NTRB gets phosphorylated, transfers phosphate group to NTRC, so both of them are now phosphorylated. And NTRC phosphorylated binds to promoter 1 side and prevents, so it does that looping, so activity of sigma 70 promoter is diminished. And in the same time, sigma 54 RNA polymerase is active, binds for second promoter and activates transcription of genes for GS. Now integrate. So these two last slides I actually added just because I thought it's going to be nice integration of both transcriptional and enzymatic content. So let's see what's happening when there is a, and again, these two slides are, you have to know them. Like these last two slides, very important. When there is enough nitrogen, when there is no starvation, so that means high glutamine, low alpha ketone. First, who is sending it? GLND. GLND has dual function, UR and UT. When there is enough glutamine, GLND starts its UR activity, which means removal of UMP groups from GLNB. The other name for GLNB is P2 product. So now GLNB is deuridylated or naked. GLNB naked interacts with GLNE, which is glutamine synthase, and it activates its it's not glutamine synthase, sorry, what am I saying? GLNE, got it, sorry. GLNE activates its AT activities, so, and adds AMP, like adenyl groups to GC, and that inactivates GC. So when GC is adenylate, that means it's inactive, okay? In the same time, naked GLNB binds to NTRB kinase domain and inactivates it, and it stimulates phosphatase activity of NTRB. NTRB cannot phosphorylate NTRC, and there will be no transcription of GS expression genes. Is that clear? Is that clear? What's happening in low level of nitrogen? In low level of nitrogen, when there is a low glutamine, again, GLND will send a low level of glutamine and high alpha ketoglutarate. That means UT activity of GLND. GLND adds uridyl group to GLNB, so it gets uridylated. Uridylated GLNB binds to GLNE, and GLNE activates AR activity, and AMP groups are removed from GC, which means GC is now active. And in the same time, uridylated GLNB binds to NTRB that will stimulate kinase activity and subsequent phosphorylation, which will now activate transcription of GNs. And this is just a beautiful picture that, if, in case you like more pictures than text, so everything what I said is here. And that would be all for today. I think it's more than enough.